And of course, we are trying to see if the power of computation can help us here, right. There may be other experimental ways of answering the question that we just posed, but we are more interested in is this question on figuring out the routes through which a cell senses and relays external signals. Can it even be answered through computation? Can computational analysis provide some hypothesis which can then be used to guide future experiments? Okay, that is the goal. And so, can a computational or algorithmic thinking help us here? Okay. Maybe it will help, but we need to first pose the exact computational problem, because all the things that I have told so far, if you explain to a computer scientist with no knowledge of biology, they will just open their hands and say that, okay, I do not understand what you mean by a signaling molecule and a pathway and map triple K, map double K, all of these are jargon that will just fly over the head, right. So, we need to be able to pose the exact computational problem, and then we need to get an exact concrete formulation. We are going to do it iteratively in the next 10 minutes or so. Okay. So, one particular computational problem, we wear our computational hats, could be that can we find all possible signaling pathways or molecular pathways hidden in a large scale biological network. Okay. And even here we need to be able to computationally describe what is a large scale biological network and also computationally describe what a signaling pathway is, so that we will be able to search for signaling pathways in a large biological network. Okay. So, let us just look at what a large scale biological network is before we go to what a signaling pathway is in computational terms. Okay. What do I mean by computational terms? I have already seen this before. You may recall that we showed this gene regulatory network, right, where a gene G1 was controlling a gene G2 and both of them were controlling a gene G3 through this series of protein products from these genes. So, and then we were able to say that this form of relations, this form of regulation of genes by other genes can be represented as a gene regulatory network, and we can use an adjacency matrix or an adjacency list representation of this graph to capture the forms of interaction and regulation among these different genes, right. So, we need to come to this level of detail if we were to pose a concrete computational problem. Okay. And we are going to work with another type of network called a protein-protein interaction network. So, this is a gene regulatory network, and then it captures some of the protein DNA interactions. So, this gene 1 produces protein 1, right, through this intermediary mRNA 1. This was the central dogma that we had seen before, right. And this protein 1 goes and binds to the DNA upstream of this gene 2 and then activates it to produce protein 2 via its intermediary mRNA 2, right. And so, this kind of protein DNA interactions specifically shown here is captured in this, or in a simpler manner in this gene regulatory network, right. So, but how can we capture protein-protein interactions, right. So, protein-protein interactions are captured in a different type of network. Instead of a gene regulatory network, we have a protein-protein network or a protein-protein interaction network, a PPI network. One particular PPI network is shown here. Okay. So, here in this particular network, all the nodes, these little dots that are shown here are different proteins. Okay. This is a yeast PPI network. So, each of these dots are different proteins. There are about 5,000 proteins in yeast, or 4,500 roughly. So, so many of those proteins are shown here, and if you see an edge or a link between two proteins, it means that the two proteins are physically interacting. Okay. So, there is some form of physical or biochemical interaction between those two proteins. Okay. And then usually proteins can interact either in pairs or in complexes, but for simplicity we are going to just represent all these interactions using pairwise interactions. Okay. But in actual biological system, proteins can either have pairwise interactions, two proteins interact with each other, or more than two proteins can also come together and interact as form of a complex. Okay. And both these types of interactions, pairwise or complex type of interactions, there are experiments that are available to screen for these interactions, to observe these interactions at a large scale. Okay. There are high throughput experiments done in yeast or mammalian cell lines, where you can observe which proteins are interacting with which other proteins. Okay. And shown in here are two such experiments, just two hybrid experiment and affinity purification mass spectrometry APMS experiment. Okay. I am not going to go into the details of these experiments, but just wanted to show this to say that you have a bait protein and a prey protein. Okay, in both of these experiments. In the two hybrid experiments, one of the protein is a bait protein, another is a prey protein, and you want to know whether there exists an interaction between these two proteins. So, this will tell you whether that interaction exists for a large number of bait and prey proteins. Okay. In the APMS experiment, you also have a bait protein, but instead of one prey protein, you have multiple prey proteins. And so, for each bait, then you can figure out in one go what are all the other proteins, the prey proteins that interact with this bait protein. Okay. But this kind of bait prey interactions, these interactions between two proteins or one bait and multiple prey proteins, right, is not a clean process. Okay. So, all of these experiments are happening, these are wet lab experiments, and there are multiple steps involved like shown here, and then there could be noise introduced in different steps along this experimental process. So, we have a lot of false positives and false negatives. So, what do I mean by that? False negatives is when you know that two proteins truly interact, but then you do not observe it in your experiments, that is false negatives, right. And false positives would be these experiments tell you that two proteins interact, right. So, maybe this particular bait is protein 1 and this prey is protein 2. So, this may say that protein 1 interacts with protein 2, but in the actual underlying system there may not be a true interaction, right. So, you have these noises or false positives and false negatives, and these are very common in these large scale high throughput experimental data. Okay. So, we need to be able to actually look at this probability that protein U interacts with protein V. So, given two proteins U and V, right, we need to be able to look at all the experimental evidence. So, there is not one study or two studies. So, there are multiple studies that have looked at all the interactions in yeast or other mammalian cell lines, right. So, for a particular pair of proteins U and V, then you can look at all the studies that have observed whether these two proteins interact or not, right, and then collect the evidence and aggregate that evidence to estimate this particular probability that the protein U truly interacts with protein V in the underlying system based on all these experimental evidences from these high throughput experiments. Okay. So, then we will be able to quantify the signal versus noise in each of these observed protein-protein interactions. If we are successfully able to do this, then we will have a well founded probability, and then we can for every pair of proteins, right, then we can make up this graph. So, we can have this graph, this PPI network G equal to V E W. V is a set of nodes or the proteins, E is the set of pairwise protein-protein interactions, W is the weight which indicates the probability of true interactions for every edge that you have observed, right. If we can do this, then we have this weighted undirected graph which we can then take forward. Okay. This is what I mean by we have to quantify, we have to make the problem concrete. So, we now have a graph that we can work with, and in this graph we are representing a protein-protein interaction network along with confidence on each interaction. So, what is the probability that each interaction in this network is actually a true interaction. Okay. I am not telling you how we are going to estimate this probability, we will get to that, but this is something that if you have this probability, then we will be able to get around the noise in the data and then focus on the more confident edges in the graph. So, that is explaining what a large scale biological network is. A large scale biological network is noisy, so we have converted it into an undirected weighted graph, where the weight will tell you the confidence in each of these interactions, the probability of the true interaction for that particular edge. Now, once we have this graph, then how do we look for signaling pathways in this graph? Okay, what signaling pathways are hidden in this graph? So, we need to now come up with some kind of a grammar of a signaling pathway. So, how do signaling pathways look like? So, what are the substructures in this graph that are corresponding to signaling pathways? To do that, we go back to a canonical example. In canonical example, you have seen that signaling pathways are these linear chain like pathways, right. So, it is a series of nodes all connected by with the adjacent nodes being connected by edges. Okay. So, there is a path that goes from a receptor protein and it uses certain other green proteins, some of them are kinases, and then it comes all the way and ends in a transcription factor or a regulator protein, right. Here are some more examples and all of them have the same pattern, right. So, the one that we just looked at is this leftmost one, the pheromone response. There are also other pathways for growth invasion, cell wall integrity, high osmolarity response in yeast, and almost all of them you can see that they all start with receptors, these blue proteins, then they have some kinases which are these, and other proteins which are these green proteins, and they end in these transcription factors. Okay. That is these red proteins that are shown here. Okay. So, that is what I mean by a grammar of a signaling pathway. Like I have said, they all start with a certain set of proteins, they end with regulator proteins, they start with receptor proteins or membrane proteins, end with regulator proteins, which are also known as transcription factors. So, those are the blue ones and the red ones, and then they have some other proteins in between. They are mostly, I mean, they are all slightly different from paths, but they are mostly chain like or linear paths. Okay. So, then this gives us an idea, right. On putting these two things together, we have a PPI network, which is a weighted undirected graph, right. So, which is shown here on the left, and then we want to find linear signaling pathways that are hidden within this network, right, and we want to come up with a graph algorithm that will analyze this network shown on the left and then figure out all these linear pathways shown on the right. Okay. And these linear simple paths are from the receptor to the regulator proteins. Okay. So, that is what we have achieved so far. We still need to make it a little bit more concrete. Okay. And what do I mean by that? You can start with this network, right, and then you can see that there may be many options for this path. There may be more than one path. So, this could be, for instance, a regulator protein or a transcription factor 1. This could be another transcription factor 2. Okay. For a transcription factor 1, maybe this is a receptor protein, I will call it R1, and then there is a nice path that goes along this way, right. So, then you have this signaling pathway that goes from this receptor protein to a transcription factor, right. But for this transcription factor 2, maybe there is no receptor proteins nearby, maybe another receptor protein is either this R1 or maybe R2 here, and to go all the way from R2 to TF2 is going to be a very long path, right, and maybe that is not biologically feasible, right. So, then for TF1 it is feasible, for TF2 it is not feasible. So, we need to know some prior expectations of how long a path you want. Okay. So, length of a path in terms of the number of nodes in a path that you desire would be good to have a priori, and then fix that as a parameter that is input to the problem. Okay. So, these are some of the details that are missing that makes the problem still vague, but we are getting closer there. Okay. So, to summarize what we have seen so far, right, we have gone from the biological question, what are all the different routes through which a cell senses external signals and relays this information to the nucleus, to these two specific computational problems, I would say, on understanding what a large scale biological network is, and then understanding what is a signaling pathway hidden within these large scale biological networks, right. And then we are still, so over here as a computational person, the computational details are very minimal here. So, if you are a CS person without any knowledge of biology, this is how you will feel after you read this statement, right, because it just cells, molecular routes, external signals, nucleus and so on, too many different terms. Here it is a little bit better, you want to find all linear signaling pathways in a large biological network. So, you are a little bit better, you are starting to be happy about this formulation, but still you are not fully happy because there are still certain details that are missing. If you do not have a concrete computational problem, you cannot solve it, right. So, that is just the fact. And so, we want to now come up and formulate very concrete problems for this 1A and 1B, right, on what a large scale biological network is, and then how do you search for signaling pathways in that biological network, so that as a CS person, a computer science person, we are happy about it, and then we can then start proposing solutions for these problems. Okay. And that is what we are going to do next. Okay. Let us look at computational problem 1A, right, to make it very precise, right. So, we have noisy high throughput experiments in which protein-protein interactions have been observed. Now, for every pair of proteins, we want to estimate the probability that those pair of proteins truly interact, okay, and that is what is given here. Your input is two proteins U and V, and then you have the list of experimental studies where this protein pair has been observed to interact, right. And then you have some additional features also. So, these proteins, you have corresponding genes which are producing these protein products, right. So, you can also have information like expression similarity of the two genes that are encoding for these proteins, and a few other input features that will tell you that will give you some indirect evidence for whether these two proteins are interacting or not. Okay. So, you have the list of experimental studies where these two proteins are observed to interact, and you have these additional features such as expression similarity of the two corresponding genes. Now your output is you want to know the probability that protein U truly interacts with protein V based on all these input features, okay, all these experimental evidences and the other inputs. Okay. So, that is the specific computational problem here, and we want this problem to work for any pair of proteins U comma V. Okay. Let us just stop and think for a bit about what approach you will take to solve this problem. Okay. So, we want to estimate this probability, and then we want to give higher weightage to increasing number of studies in which this pair of proteins have been observed. Okay. And I can come up with any kind of ad hoc formula. If the number of studies is at least 5, then I will have a probability of 1, and if the number of studies is less than 5, then I will have a probability of 0, but then you may also want to not have this binary 0 or 1, you may want to give some intermediate probability values. And so, you can see that any ad hoc approach is not very fully satisfying, and if you put on your ML hat, machine learning hat, you will see that if you have a training set of true interactions and kind of positive examples and negative examples, right, then you can try to come up with a more careful way of estimating these probabilities. Okay. And that is what we are going to add as a footnote here, right. We prefer an ML, a machine learning approach to solve the problem. For training this model, we need access to a ground truth set of interacting like positive class and non-interacting negative class set of protein pairs. Okay. And hopefully you can now see how the ML model naturally comes in. You have a binary classification model. For every pair of proteins, you need to predict whether it will interact or not interact, and that binary classification model, many of the ML classification models comes along with a 0 to 1 score, right, that can be interpreted as a probability of the positive class, right, probability of an example belonging to the positive class. So, that is what we are going to use. Okay, that kind of approach is what we are going to use. Once we have that probability, then we can just apply it to every pair of proteins, and you can just get this graph, right, where we said this V is the set of proteins, E is the set of all the protein-protein interactions, PPIs, and W is a function which maps from each edge to a real number between 0 and 1, and this weight of each edge is going to be the probability of the edge estimated based on the problem 1a formulation. Okay. And so, then we are going to use this graph to define our problem 1b, which is looking for signaling pathways in this graph, right. And before we go there, let us just make sure that we understand these edges carefully. So, if you have an edge E connecting two proteins U and V, so weight of the edge or weight of U comma V is going to be the probability that protein U interacts with protein V based on all the experimental evidences. So, this is the probability that is returned by solving problem 1a. Okay. Now, if you have these weights, let us check our understanding of how these weights can be put together to weights of individual edges can be put together to find the weight of a path, okay, path consisting of a series of edges. So, what is the probability of a simple path of length k in the PPI network G? Okay. So, let us just look at this graph G. You have the set of proteins and all these interactions, and you are looking at let us say a path of length 3, sorry, V1, V2 and then V3, okay, and then this edge is going to be called E1, this edge is going to be called E2. Okay. So, you have a length of a path here is 3, we are going to follow the convention that the length of a path is the number of vertices. So, in this case your length of the path is k equal to 3 nodes in this paths V1, V2 and V3. So, the length is going to be 3, and then there are two edges E1 and E2. E1 is the edge which connects V1 to V2 and E2 is the edge which connects V2 to V3, right. So, now what is the probability of this path E1 comma E2? We can just assume that they are independent. It is not always the case that they are independent. We can assume that they are independent, and we can say that this is going to be probability of E1 and times probability of E2, right. Probability that the V1 and V2, those two proteins truly interact, times the probability that V2 interacts with V3, right. And that is what is essentially shown here for a general k length path. Okay. You have probability that path P equal to V1, V2 etc. to Vk is a probability of E1 equal to V1 comma V2 times probability of E2 equal to V2 comma V3 times probability of Ek minus 1 equal to Vk minus 1 to Vk, right, and this is the product of each of these individual H probabilities. Okay. We are also asking for a simple path of length k. A simple path, if you may recall from your graph theory, is a path which does not have repeated vertices. So, therefore, it will also not have repeated edges. It will visit different vertices only once, it will not come back to the same vertex. Okay. So, it will have a set of distinct vertices in the path, that is called as a simple path, or sometimes we just ignore the simple and just call it as a path. Okay. So, paths are simple, they do not have repeated vertices. Okay. So, none of these V1, V2, Vk repeats again, right. So, they are all distinct vertices. So, this is the probability of a path assuming that the edges are all having independent evidences, right. It is not necessarily true, but we will make that assumption. Now, this is a multiplicative score for a path. So, from the scores or probabilities of the edges, we have obtained the probability of a path which is a sequence of edges, right. So, sequence of edges in the graph, a series of edges in the graph. But many graph algorithms actually work with additive scores rather than multiplicative scores. So, how do you convert this multiplicative score into an additive score? A standard trick, you log transform the probabilities. So, then the log of this left hand side, log of probability of this path becomes log of the right hand side, which will be log of probability of E1 plus log of probability of E2 and so on, plus log of probability of Ek minus 1. Okay. So, then this is now along with the convention of traditional graph algorithms which like additive scores, right. So, then we can redefine your weight of edge or weight of u comma v to be not just the probability that u interacts with v, but the log of the probability that u truly interacts with v. Okay. So, then a score of a path is going to be the sum of the weights of all edges in the path. Okay. That is a nice convention to follow because the graph algorithms are generally prefer that convention. Okay. Now that we have that set straight, we are now ready to define problem 1b. Okay. The computational problem 1b is a simple path search problem. You want to find the best scoring paths of length k in an input graph. Okay. And your inputs are G, which is this representing a PPI network, and the W is the one that is returned by problem 1a, right. And then you have a starting set S contained in V, which represents the receptor proteins or membrane proteins where you can start the signaling pathway from, right. And then you have a terminating set T contained in V, which represents the regulatory proteins, right. Regulator proteins are transcription factors, right. These are the final set of proteins where we want the signaling pathway to end. And then we have a certain path length k. So, typically in yeast they set k to be 5 to 10 and so on, right. So, we have a small pathway length that is biologically motivated. So, we can just get that also as an input from the user. Okay. Now, what is the output that is desired? For each node t in T. So, for each of the terminating set node, I want to find the highest scoring simple path of length k from at least one starting node. So, from some starting node S in S to the node T. Okay. So, if I were to draw it like a diagram, I have this overall graph G that is provided as an input. It is a weighted graph, undirected graph. The weights are the log of the probabilities of proteins interacting amongst themselves, right. And then you have a starting set S, which is all of your receptor proteins, and then you have an ending set T, which is your transcription factors or the regulator proteins, and you are looking for a path of length for every T here, right. You want to know what is the best path from some node S in S, small s in S, and of a certain length. If you are looking for length 4, right, if k equal to 4, then this will be one path. So, the S is the first node, this is the second node, this is the third node, and T is the final node, the fourth node. Okay. So, this is really there may be many paths from here which are of length 4, right. I want the best such path, maybe that path is over here, this path may be the best such path in terms of the score of the path. Okay. The score of the path, again to remind you, is the sum of the weights of the edges along the path. Okay.